my name is David Finlay, and I'm a biochemist. I'm an immunologist, depending on who I'm talking to. So I wasn't sure when I was asked to give this talk, I wasn't sure what sort of audience to pitch it to, so I'll kind of pitch it to the general audience, general public, plus a bit of extra complexity. So, I think perhaps we all have a bit of science background here. Anybody here has no science background at all? You're not all professors, are you? Okay, so I think we'll be fine. So it is, but even still, metabolism tends to be one of those subjects that people, they may have learned back in undergrad and they go, oh, is metabolism awful? And they throw the big arguments. So we'll go through slowly. So there are some more complex slides, but in general, I'd pitch to a wide audience, given that you may be chemists or immunologists or biochemists or biologists or whatever expertise you've got. Okay, so do shout out if you think I'm being too detailed or not detailed enough. So, does everybody know who this guy is? Can you recognize him? So, this is James Allison. So usually somebody would recognize this. He won the Nobel Prize 2 years ago. 2 years ago, along with classical uncle. And I think he's a good example. So everybody, when they stand up and give a science talk, it's usually a nice story, and it's like, I'm going to cure cancer. And might also be something similar, but everybody always has a nice story. But the reality is, a scientist, and I think there's one of the grad scientists here, that's not how science works. And I love science, I love being a scientist, but it's not. I didn't set out to cure cancer, and I'm not curing cancer. I set out because I was just curious. I'm really curious about how the world around me works. I particularly like biochemistry. And I go to work every day asking little questions, and we design an experiment, and we get little answers, and they're only kind of little pieces of the jigsaw. But then when you see people giving talks like this, they tend to be a real nice story, because we all like stories, and then in the end we all do them together. But it all starts out getting those little tiny little jigsaw pieces of little clues of what's going on. And often, the final piece you get could be the first result you got, or the last piece of data I show you could be, or the first piece of data could be the last piece you get. It all kind of gets put together at the end. But scientists don't go out to generally cure diseases, they go out to find out how things work. And James Allison had a great quote, because he got the Nobel Prize, the first ever Nobel Prize for a cancer therapy. So this is a big thing. All the cancer research has gone on before. This is the first research prize for cancer therapy. So you thought, with this guy obviously would never cure cancer. But the first thing he said when he was interviewed was, I'm a basic scientist. I just wanted to know how T cells work. It's as simple as that. He just wanted to know how it worked. And he said he was very lucky that after 20 years of trying to figure out how T cells work, he came across something that was then harnessed, and it's the biggest breakthrough in cancer research that we've had in decades. And his work led to checkpoint inhibitors, which are now used as almost like a routine for cancer therapy, and they can have some absolutely spectacular results in some patients, not all patients, but it really was the first time where we really truly successfully able to harness the immune system to treat cancer. But he just wanted to know how T cells worked. So I'm going to tell you a story about what fuels killer cells, in particular natural killer cells, and what our work has done to maybe suggest ways forward in harnessing these as a therapy. But in reality, I just wanted to know how NK cells work. And we'll go through that as we go along. And it's another quote from another famous scientist, and maybe he was again saying how scientists, we are in general curious people, and I am curious. That's why I love it. And maybe I am a bit of a child, but I think his point was that that's one thing scientists have in common with the children, is that we maintain our curiosity, and we just want to know how things work. And I hope if anybody's going into a career in science, that's one thing you need to grasp on, to be curious, and also always question things. Even if it's in a textbook, it means there's a lot of data support there, but sometimes textbooks aren't right either, because everything can be good. As a scientist, you should be learning how to question everything. And this is also unfortunately his curiosity led him to do the groundwork for making an atomic bomb, which maybe wasn't quite so good, but curiosity still is an important characteristic to have as a scientist. But I just want to start out with that sort of general thoughts on being a scientist and what led me into this sort of career. So killer cells. These are natural killer cells, and they are called killer cells because they have an innate ability to kill tumour cells and virally infected cells. On the left, you can see there's a tumour cell, the elongated one, the tumour cell. This is just this imaging in a test tube. You can see a natural killer cell coming along here. It takes a good look at the cell, and then shortly after, you see the red dye is a dye that's showing you the cells engaging in apoptosis. So here you see it's recognizing it and it reduces apoptosis, and then a little time later you see it's shriveled up. Here's another example of a tumour cell, and these very highly motile natural killer cells sort of searching out for target cells. When they find one, you can see apoptosis being induced here. There's lots of nice images of this. Here's a real nice image of multiple natural killer cells surrounding. Actually, these are actually killer T cells surrounding a target cell. You can see how they kind of make very close distance, kind of just making first contact. This one's making slightly more contact. And here you can see this cell is really having a good look at this cell. It's really is probing and looking at that cell and seeing whether this is something you want to kill or not. Obviously, if you have the ability to kill cells, you don't want that to be unleashed without due consideration and good thought. So it's very important that they decide on what to kill very carefully. There's a lot of very careful mechanisms to ensure that. And you can see here we've stained for, you can see this one's forming a nice synapse. The concentration of the green here shows that it's really inspecting it. This is the actin cytoskeleton skeleton forming a synapse. And in red here is stained the cytotoxic granules, and they are what they sound. These are sort of basically small little parcels of toxic molecules that killer T cells and or natural killer cells can deliver to a target cell. So once it decides it wants to kill it, it will deliver these cells. Here's some movies. So here again is a killer cell. You can see how closely it is inspected. It's really given a good hope there, you know. So there's a huge amount of shared space there between the cells where it can inspect the target cell. Same here. You can see this really close synapse. See the sort of ring that's formed around it? It's really attached to the cell, looking at all the attributes on the surface of that cell, deciding whether it is a normal healthy cell, in which case you should leave it alone, or whether it's a target cells in particular, whether it's a virally infected cell, in which case it needs to be killed, whether it's a transformed cell, cancer cell, in which case it probably has lots of stress molecules on it. That's how it identifies as abnormal, in which case it needs to be killed. And you can see once it makes a decision, these in red, these cytotoxic molecules, they travel from the center of the cell all the way to the membrane, where they are then basically delivered into that site, into the space between the target cell and the natural killer cell. And these are little parcels of toxic granule. They puncture holes in the target cell. They allow enzymes into the target cells and then start chopping up all the DNA and inducing apoptosis. It's really potent. But you can see there are cellular assassins. They're much more. They're very specific. They aren't just random killers, you know. So they're very much. They're picking and choosing their target cells very carefully. They're not indiscriminate killers, and of course that's what you want, because you don't want to be damaging healthy tissue. You don't want to be damaging all the non-virally infected cells. You want to be really specifically targeting the ones that are virally infected or the ones that are transformed. And so they are very, as you can see by these moves, very careful. They're very careful in what they kill, and they are important cells. When we talk about natural killer cells, there are a few varieties of killer cells. We have our cytotoxic T cells, which are also killer cells. But we've been working mostly on natural killer cells for the last 7 or 8 years. And these are important cells for protecting us from cancer. They are also important for protecting us from virus. And more so, there's a sort of quite a lot of hope that these natural killer cells could be useful to harnessing their ability to detect and kill tumour cells for types of immunotherapy. So can we harness the power of these cells to provide new and better ways of targeting and killing tumour cells within patients? So, you know, essentially what you want to have is, in a killer cell, you want to have the ability to really pick out this one transformed cell or a cluster of transformed cells amongst normal healthy tissue. And this is particularly when you think about sort of when the cancer starts to spread to other sites in the body away from the primary tumour. This is called metastasis. And so you really there's a hope that natural killer cells could be particularly useful in finding these sort of micrometastases and these tumour cell clusters. and specifically finding them and killing them without damaging normal tissue. So there's a lot of hope in immunotherapy ever since James Allison had his really great discovery of finding checkpoint inhibitors that have the ability to really reactivate, rejuvenate the immune system, to allow the immune system to detect and eradicate cancer. There's lots of hope, and we can find new and better and more efficient ways of harnessing the patient's own immune system to fight cancer. And NK cells could be important in this. Actually, they already are important for certain therapies. So, for instance, you may well have heard of Herceptin. Herceptin is a treatment that's given for breast cancer. It's basically an antibody. So, one of these little molecules here, an antibody. And what this does is it's got a specific domains on the antibody that recognizes the estrogen receptor on the HER2 positive tumor cells. So they get injected in and they code the tumor cell. Putting an antibody on a cell doesn't kill the cell. You need natural killer cells to come along, and natural killer cells, one of their triggers is if they see an antibody-coated cell, they don't think twice, they just kill that cell. It's called antibody-dependent cytotoxicity, antibody-dependent killing. So if ever is the case that natural killer cells comes along and sees an antibody-coated cell, it just kills it. In this case, it doesn't think twice about it. So in the other examples, we showed you how it carefully inspects the cell. If it's antibody coated, it just kills it. So NK cells are important for for how well this treatment works, the efficacy of Herceptin. So if your NK cells don't work, it means that even when the antibody gets into these patients and coats the tumor cells, the killing of those cells isn't very efficient. For a long time, people have been trying to use NK cells as an adoptive transfer therapy. Adoptive transfer means you take the cells out of the patient, you grow them up in the lab, you activate them so they upregulate all their killing machinery, they have more and more of those cytotoxic molecules, and then you stick them back into the patient. So you can either do this taking cells from the patient, or you can take cells from donors, and you're sticking millions and millions of these cells in, fully activated and ready to go find and kill the tumor. These are NK cell based therapies. These have shown quite a good bit of success in treating sort of liquid tumors like lymphomas and leukemias. And people are trying to find out ways to make them work even better to try and target solid tumors like breast cancer and other solid tumors. And that's a lot of work still to do there. That's just basically cellular therapies, taking NK cells from a donor or from a patient, expanding, activating them, and then putting them back in again. And there's also a lot of interest in engineering cells. So you may have heard of CAR T cells, which is it's engineering a T cell for to specifically recognize the tumor you want, and then growing them up in the lab. And engineering suggests what you might think is. Basically, you're trying to think what sort of attributes do I want in that cell, and we'll engineer that. So you start engineering using recombinant DNA technology, and you can stick on that receptor, you can stick on that molecule so it recognizes the tumor better. Basically, engineering. People are working on CAR NK cells as well. So these are starting with an NK cell and sticking on a receptor that recognizes something on the tumor, and modifying and engineering that cell so that it's better able to find and kill tumor cells when they get into the patient. So there's a lot of work going on this. People are working on trying to find so-called off-the-shelf therapies. So they're trying to find a way of engineering these NK cells and then making huge amounts of them, freezing them down. So when patients come in and with a certain cancer, they can just go to their liquid nitrogen, their freezer, take out the vials, thaw them out, grow them up, and stick them to the patient and provide therapy, because having that sort of off-the-shelf therapy makes it much more feasible, much more economically viable to treat patients with these cellular-based therapies. Because the reality is that the cost involved in taking the cells out of a patient, growing them up, putting them back in, it's a very expensive business. But these costs are coming down a lot more, and as they come down, these approaches become more and more viable. So there's a lot of interest in NK cells, for cellular cancer therapy. So, in a healthy person, NK cells are able to recognize a tumor cell. They have multiple ways of killing them. So I've told you about these granules here, or these packages of toxic molecules that they can exocytose directly at the tumor cell. They puncture holes in the tumor cell, and they let these enzymes called granzymes into the cell that trigger apoptosis or program cell death and kill the tumour cells. That's not the only way they can kill them. They also have cytokines that they can secrete that can have anti-tumour functions. They have death receptor ligands that they can express. But they're also important beyond just directly targeting the cell. They're also important in orchestrating the immune response. So they also release other cytokines. Cytokines are basically just signaling molecules that sort of intracellular signaling molecules. So cytokines like interferon gamma, which are released by NK cells, can then communicate to other immune cells, tell all the immune cells to come and bring the immune response to the site of the tumour. So basically, coordination between the different cells of our immune system. So this, in a healthy person, this lots of important functions of an NK cell: direct killing, making cytokines, alerting other cells of the immune system, really getting the whole anti-tumour response going. Unfortunately, in cancer patients, it's now emerging that not only do they have cancer, but the cells that are in their bodies that are supposed to be fighting the cancer don't work very well. So we've looked particularly in breast cancer with our collaborators Claire Gardner in Dublin and other collaborators in various hospitals in Dublin, and NK cells just aren't working. So the NK cells, they don't make as much cytokine, they don't kill as well. So this is a problem. So especially if you think about patients receiving Herceptin, where you're given these antibodies that are going to coat the tumour cell. But if the NK cell that's supposed to come along and kill that tumour cell isn't working very well, that's not so good, is it? So we really want to understand, why they aren't working and how we can make them work better. And also, if you think about why they're not working, well, if you're going to then generate these expensive cellular therapies where you're going to inject NK cells into a cancer patient, you don't want them going into the cancer patient and then being turned off for some reason and becoming like those NK cells I just showed you, which aren't working. So we need to understand why in the environment within a patient with cancer is such that the NK cells stop working, stop having the optimal anti-cancer function. So this is a big caveat. So NK cells are really exciting in terms of cancer therapy, but they do not work very well in cancer patients, and we want to know why. So what we approach it slightly different maybe to a lot of people working on NK cells. I'm not a proper immunologist, I'm probably not a proper biochemist either, and I'm a half-breed. So I often show this slide, and I think I offend everybody in the audience because I offend biochemists and immunologists alike, because I use the analogy of a jack-in-the-box. So I think of biochemists and immunologists. Immunologists really care about what comes out of the cell. So in terms of immunologists are all about, if it's a T cell, it's what cytokines are they making? Oh, they're making IL-17 and Th17 cells. So immunologists are obsessed with what comes out of the cell, but they don't really care what happens if, are there any immunologists here that are really offended? I apologize in advance. So this is a very reductionist approach, but immunologists in general like to understand what cell it is, what comes out of the cell. But they aren't so worried about the mechanisms that are happening within the cell. But a biochemist is obsessed with the mechanisms. So if I was a biochemist here, I wouldn't be caring about the fact that something's jumping out here. I want to know how the mechanism works, what's the trigger in there, and all those sort of things. So because I guess I've got a background in both, we have a slightly different focus. So an immunologist will probably say that I don't do immunology properly, and a biochemist often says that I don't really do biochemistry properly. But I think we kind of come there in the middle, and we do something slightly different. So there's the immunologist and there's the biochemist. So essentially what we want to do is we want to look at metabolism, and we want to look at the immune cell function and the ability to kill tumours. And when I think about the anti-tumour NK cell response, I think about a car and a car analogy. Okay? So and the same for I guess the T cells. The T cells are another important cell that are being harnessed to kill tumours. So if you think about our anti-tumour immune response, it's like a car. James Allison discovered checkpoint inhibitors, which can be absolutely miraculous in a way they can cure cancers in some patients. So people have been given checkpoint inhibitors who have stage 4 melanoma on death's door, and they have a complete recovery. Their immune system completely is reactivated, and it comes along and absolutely eradicates all the tumour cells. That works. Maybe 15% of patients get a good response, but that's not not satisfactory. You want to treat all the patients. So obviously there's a lot of work to do, and a lot of work to understand why it only works in those patients. So what are checkpoint inhibitors? Well, basically, checkpoint inhibitors are basically taking the brakes off the car. That's all it is. And the way it works is, so tumours have these receptors, they express these receptors called PD-L1, which binds to, so this is a ligand, it binds to a receptor on the T cell, and this can also be expressed on an NK cell, PD-1. And this is a negative, so this is a way of turning the T cell off, turning the NK cell off. There are also different brakes, there's a range of these sort of checkpoints, receptors on NK cells, not just PD-1 now, but these are basically just brakes. And you know, no matter if there's a way of turning the immune system off, tumours will find it. They evolve to find it. So it's not surprising they found that engaging this receptor is a way of turning the T cell or the NK cell off. So it's a brake. And those brakes are important in our immune system because our immune cells, these cells are cytotoxic cells. They can kill the cells very, very efficiently. You know, our immune systems evolved to have brakes because we need to keep things in check. If our immune system goes out of control, starts killing a whole range of tissues, of cells, you end up getting diseases like autoimmune disease. You end up getting pathology. The people who die from the flu, they don't necessarily die from the virus. They die from the immune response to the virus. So it's the T cells not becoming overactivated, and that's what causes pathology. So we evolved to have these brakes for good reasons in terms of infection, in terms of our infectious responses to bacteria and viruses. But tumours then also are very good at hijacking these systems to find ways for themselves to survive by turning the brakes off. So basically, what the big discovery by Jim Allison and Hongzhou was basically that if you block this receptor, you can reactivate the T cell and then kill the tumor cell. So basically, you're basically inhibiting an inhibitor, turning the brakes off. So go back to our car analogy. Well, that makes some sort of sense, yeah. If you take the brakes off, that would make the car go. Well, no, it would make the car go if it's parked on a hill, and that's fine. Maybe the car isn't parked on the hill. So taking the brakes off will make the car move forward if it's parked on the hill. But maybe those patients that don't respond, there's other things that need to be considered. So, for instance, do you need to stick your foot in the accelerator as well? So if you're not parked on an inhibitor, if you're not parked on a hill, taking the brake off doesn't do anything. You also got to give it a bit of gas. So lots of people are working on that. So are there ways that we can actually find different ways to actually give the NK cells, the natural killer cells, their killer cells a bit of a stimulus, a bit of a kick, so that in combination with taking the brakes off, you can get them to start working again. You can get to find those tumor cells. You can get them to kill the tumor cells. So that's not what we work on at all. So that's a really exciting area, and that's certainly very important. What we work on is, again, taking the car analogy further, is there's no point taking the brakes on, there's no point putting the foot in the gas if your engine isn't turned on, or if you've got no fuel in the tank. So this is what we work on. We work on cellular metabolism. Cells can't do things unless they have the energy to do it. They also need to build things. So if you think about your natural killer cells, what are they trying to build? Well, they're building those cytotoxic molecules. If they're going to keep on killing, they need to make more of those toxins. They need to make more of those cytokines. So for an effective killer, you need to have plenty of energy, and you need to be able to build lots of stuff, lots of molecules to keep the response going, to keep the ability to kill tumor cells, to keep the ability to communicate with the rest of the immune system. So until recently, I guess in the last 7 or 8 years, people haven't really looked at metabolism of immune cells much, but it's becoming an emerging area of interest. So we want to know what's fueling this. So this natural killer cell, what does it need as a fuel? What are the key switches within that cell to turn it on to allow it to be an efficient and persistent killer? And we reckon if we know that, we can have some clues as to why they are being inactivated in a cancer patient. If we know why they're becoming inactivated, then maybe we can start to devise therapies for turning them back on. So, what fuels them? So what's the key energy source? Well, that's one big question. And the second big question is, if you're thinking about these assassins, what are the key building blocks it needs to build the weapons that these cells use to kill tumor cells? So what do they use to make energy, and what do they need to build things that they need to maintain their responses? So in terms of energy, so anybody's done a bit of biochemistry, biochemistry will know that in the cell, our energy comes not from electricity, but from this molecule here, ATP. And this is the structure of the molecule. It's got these 3 phosphates here. You don't need to know too much about it if you're not a biochemist. But the key thing is that this last bond here between these 2 phosphates is a high energy bond. And it basically holds lots of energy in there. So when you make ATP, you make ATP with this high energy bond. And if you break that bond open, you can use the energy to do work. So in the cell, that could be to transport something from one place to the other. It can be to engage signaling communication within the cell. So this is your basic unit of energy. And I like to compare it to a glow stick. And you may have come across, if you've got kids, you might give the glow sticks. If you're young and you've been to any raves in Cambridge, you probably had glow sticks, you're waving around with your whistles. But basically, glow sticks, they don't do anything until you crack them open. So basically, they contain the energy, but they're not using it. When you crack them open, the energy is released as light. ATP is similar. It's got energy contained within that final high energy phosphate bond. You can break that bond and use that energy to do work in a cell. So we're going to talk about how cells can make energy, and we also going to talk about how cells can build. So in terms of a natural killer cell is it can recognize and kill target cells straight away. But if you give it cytokines are these signals that can be found in response to immune response. Cytokines such as IL-2 or IL-15 and various interleukin cytokines, they become much better killers. So here's a tumour killing assay where we take in natural killer cells, we put them with tumour cells, we measure how good they are at killing cells, and you see if you activate the cytokine, you can get them to kill twice as effectively. And certainly when you do this, this is associated with a change in their metabolic machinery. So I think this is my very basic. We will get a bit more complicated on this, but this is my basic diagram. Here's your NK cell. Obviously, the key things it needs is fuel. How does it use that to generate energy? And how does it do that to increase killing? So certainly, when you cytokine activate a natural killer cell, they become bigger and they increase their metabolic machinery. This is the machine we use to measure the metabolism of a cell. So this is called I don't know why they call it a seahorse. I have to have this conversation with audiences every time. They call it a seahorse. It's not nothing to do with a seahorse, but I've no explanation for why it's called a seahorse. But it is a very useful piece of kit if you want to measure metabolism, because it's rather a simple machine, but also very complicated, and allows you to measure and get graphs like this. So I'll talk to you a bit more detail about what this graph means, but this is measuring 2 different types of metabolism. This is glycolysis, which we'll discuss in a minute, and oxphos. This is the reason why we all need oxygen, because every cell uses this process called oxphos to make energy. And keep it nice and simple. First of all, you can see as you cytokine stimulate an NK cell, both these types of metabolism increase substantially, and you see that cells go from this quadrant, which is kind of like quiescence, not doing much metabolism at all, to this highly energetic quadrant here, where they're doing lots of both these types of metabolism. So it's very clear that when you activate natural killer cells to become a better killer, it's associated with these metabolic changes. And these are important, because if you just throw a spanner in the works, for instance, so if we just use a metabolic inhibitor, there's various ones we can use, and we've used lots and different ways to do this. But if you cytokine stimulate your NK cells, make them really good killers, and then stick in a metabolic inhibitor, whether it be a metabolic inhibitor of the glycolysis we talked about, or oxphos, or anywhere along those pathways, you abolish their enhanced ability to kill. So you really need to have this metabolic response to run a really efficient cancer killer or cancer cell killer in these assays. So that was very interesting to us. It was very clear that metabolism is important. So as a biochemist, we wanted to know a lot more detail. Maybe some people might be happy enough with that. We wanted to know exactly what's going on in that cell. What pathways are they using? So we did some very complicated experiments using mass spectrometry and tracing of heavy labeled molecules and all these sort of things, which I'm happy to talk to anybody if you want to know more detail. I won't bore you with the details right now, but we essentially wanted to know what fuels they were using, and it became very clear quite early on that they were primarily using glucose. So the thing with these fuels, fatty acids come from fats, and usually comes from protein, and glucose from carbohydrates, and these can all feed, and different cells use different fuels, but certainly our activated natural killer cells that are very efficient at killing we're primarily using glucose. And glucose is obviously a very important fuel source in our bodies. It's one that's very, very well regulated. Our bodies are always very good at keeping glucose levels pretty constant. So when we looked into this, we could see that glucose was being metabolized on this pathway called aerobic glycolysis. So we get the glucose converted to lactate. This is a mitochondria. You may have heard of mitochondria. These are termed the powerhouses of the cell. The mitochondria also, so once glycolysis converts glucose to pyruvate, and then there's two fates at the end of that. It can either be converted to lactate, or it can go into the mitochondria, where it can go to this process called the TCA cycle. Don't need to worry too much about the details. And engage in oxphos. This is the process in your body that uses oxygen. This is why we breathe oxygen. Every cell does this, and this gives you energy. Okay. So glucose to lactate gives you a little bit of energy. 2 molecules of ATP, not very much. It's rather inefficient. But it can be very, very fast. So it's very useful for cells that need energy very quickly, because you can ramp this up to very high speed. This is much slower, but much more efficient. So oxphos can convert glucose into 36 molecules of ATP. So very efficient in making energy. So this is your sort of biochemistry 101 for cellular metabolism. It's not just glucose, so at various points other fuels can feed in here as well. Amino acids can feed in here. Fatty acids can be broken down in the mitochondria to feed into the TCA cycle. Usually is a particularly important fuel for feeding into mitochondrial metabolism. Amino acids can also feed in here. So it's not as simple as I've shown you, but for actual immune cells, glucose does seem to be a really, and especially for killer cells, cytotoxic T cells and natural killer cells, glucose does seem to be a really important fuel. And again, the reason why you upregulate these pathways when you activate these killers is because simultaneously you can make energy, but you can also siphon out. So if you have a high rate of flux through these pathways, you can sort of divert some of the intermediates into these other pathways that are used for making the building blocks of the cell. So natural killer cells dramatically ramp up their metabolism so they can make more energy. But if you've got high levels of all these intermediates of these pathways, you can divert them into these biosynthetic pathways, and you can make proteins. Proteins are really important for the effector molecules. So granzyme B, which is one of the toxic molecules that natural killer cells kill. Cancer cells is a protein. Nucleotides are important if you want to undergo division. So an important part of the immune response is to divide. Cells can divide from one natural killer cell to two natural killer cells. So basically building an army of killers. So these are all important building blocks. Okay. So cellular metabolism can be ramped up to provide energy and to provide the building blocks of the cell. So mitochondria, mitochondria is the reason why we use oxygen. Mitochondria convert oxygen into water, and this is really important in our cells. So we focus on this a bit more, and here, so this is the simple view. You could break it down, and if you had a few hours to go with the biochemical reactions, you can break it down into a lot of different reactions, a lot of different enzymes. So it's really quite complicated. But we don't need to get into too much. But here's an important part. Here's oxygen. This is why we all breathe oxygen, is because oxygen is required by this step here in oxphos, which is required to make all this ATP. Now it looks very complicated, and if you're a biochemist, you maybe go, yeah, that makes sense. If you're a chemist, you're going, oh, maybe you're getting a bit fed up with this. Immunologist probably as well. So we'll break it down into a very simple terms. So we'll focus on this. Without any details, this is basically kind of like a power station, kind of like a hydroelectric plant. So what you have is that the mitochondria is this very nice organelle, which is multiple membranes. And essentially what you have here is a cellular membrane, which is a barrier. And the first step you have is the electron transport chain that pumps protons across, generating this big pool of protons into membrane space. And then your ATP, this really nice complicated enzyme that makes ATP, is just using the protons as a force. Okay, so you have a pump here. So your glucose is fueling these pumps that are pumping these protons across. And then here's this elegant machine here called ATP synthase. The protons come back through it, cause it to rotate, and this allows you to fuse the phosphate and ATP to make ATP. So basically, it's a very elegant way of making ATP. And you can think of it like a hydroelectric plant. So you have, in any hydroelectric plant, you have the water that drives downhill through the turbine to make electricity. This is one here. Well, the reality of this is kind of like one of those unique type of hydroelectric plant. We have one in the Dublin mountains called Turlock Hill. This is it here. It's a man-made thing where you've got a lake at the bottom, a lake at the top. And during the night, they actually pump the water up the hill, up into the upper lake, using all the cheap electricity when everybody's asleep. Electricity is not being used, excess electricity at night. They pump it up. So that's your glucose pumping those protons across. And then when they need electricity, they reverse it around and they put the water goes downhill and generates all that electricity to boil those kettles when there's an ad break in Love Island or an ad break in whatever the most popular TV program, whenever you go down and turn the kettle on. Maybe it's Irish thing, you better put these. But anyway, so it's essentially like that. You have these pumps pumping the protons across, and they have a force that drives this elegant machine that makes your energy. Okay, so that's the basics of how biochemistry works. So how do natural killer cells do it? This is where where it gets a bit interesting, because once we start understanding how this works in the natural killer cells, we can start to understand how could we go wrong with cancer and what sort of tweaks we could do to try and make it better. So the biochemistry lectures are it's a bit boring, but to understand that, we then get to understand the interesting bits of the NK cells. So to summarize our work, we found that in natural killer cells, they are really reliant on glucose. So glucose is a really important fuel. It seems to be the important fuel. And the reason for that is that here's again, going back to the simplistics, you've got your pathway, your glycolysis, your TCA cycle, then making your energy here with your hydroelectric plants here with AT synthase. So textbooks always will tell you, this is another good reason why we don't always believe the textbooks too much, always question things. When we first got these results, we were rather puzzled and we started questioning the textbooks. So in our natural killer cells, they don't really use this TCA cycle. So TCA cycle is a bit that links the glycolysis to the oxygen-consuming process that make ATP. This TCA cycle, which basically generates electrons that go into the electron transfer train and drive this whole oxphos process. In natural killer cells, they don't use it. They use something different that's called the citric malate, which is slightly different, which can also be used to make energy, but it is different, and because it's different, it's controlled by different molecules, and that's been quite interesting, and has interesting ramifications, which we'll go through in a minute. So for anybody who's got a bit of biochemistry, I'll talk you through what the citric malate is. If you get lost, it's alright, because we'll come back to the simple bits in a minute. Essentially, it starts out again with glucose. So citric malate is a metabolic configuration that's driven almost exclusively by glucose. So glucose are really important fuel for NK cells. That becomes important because glucose is one of the few things that's actually quite limited within tumours. So that's really quite important consideration. So here your glucose is converted by glycolysis. You generate acetyl-CoA, which enters into the TCA cycle, and the first thing is made is citrate. Normally this goes around, other intermediates around this TCA cycle. If you remember your biochemistry lectures, you'll remember that. But in the citric malate cycle, this doesn't go around the TCA cycle. Instead, it gets straight exported again. So pyruvate comes in, and then citrate gets exported, and then it gets metabolized into cytosol, back into malate, comes back in again, and you get this kind of weird. So it is a cycle. It's a cycle that spans the mitochondrial membrane. So it starts in the mitochondria, goes back out again, goes back in again. So it's a funny variation of it. And it does generate these molecules called these electrons. This is basically an electron carrier. So it does generate electrons to fuel this pump which we talked about called electron transfer chain. It generates these protons that then drive this generator here to make ATP. So it does the same thing that TCA cycle does. It's not as efficient, but it does do it. And so why is it different? Well, it has an additional output that it can generate things that are important for keeping glycolysis going. That this molecule is important for keeping glycolysis going fast. And it also makes this other molecule in the cytosol that the TCA cycle doesn't make, which is important for building. So it simultaneously allows you to make energy and make these building blocks that are important for making lipids and important for separation. So which could be key to its function. So that's the detailed biochemistry of what the citric malate is. And why is that important? Well, finding that out, this novel configuration allowed us to ask, well, what is allowing the NK cell to turn that on? And we found a switch, a novel switch, which is very unexpected, called STREB. STREB is a transcription factor, and basically this switches on this novel configuration called the metabolism. This was surprising. We didn't discover STREB. STREB's been discovered and it's been being studied for a long, long time. There's been Nobel Prizes around STREB and its role in controlling cholesterol metabolism. And it's the master regulator for cells to make fatty acids, to make cholesterol, so it's very, very important. But this is virtually the first time we found that STREB is important to making energy, so it's really quite interesting and novel. And of course, as soon as you find the key switch, you may have found a key reason why something is switching it off. So if we can switch that on, it's quite possible that tumours can switch that off. And we've shown that if you switch this off, it really has disastrous consequences for the metabolism of the NK cell and their ability to kill tumour cells. So we started out thinking that we're going to be looking at a cell which is upregulating metabolism to make ATP through the TCA cycle. And what we found was they were doing a different configuration. And the reason for this was because of STREB and the importance of this transcription factor called STREB in these cells. So I just want to show you some data confirming this. This is coming back to our seahorse machine that measures metabolism. So I showed you a really quite complex metabolic pathway here. Seahorse is quite simple because it just measures two things. It measures lactate, which is basically an acid. It measures acidity. And lactate is pretty much the only. So when a cell makes lactate from glucose, it kicks this lactate out of the cell. So this lactate ends up outside the cell. So you can measure extracellular acidification. So basically, it's just measuring pH. It's pretty simple. The other thing that it measures is the two probes. It also measures oxygen levels. So by measuring pH and oxygen, you can get a real nice measure of these two pathways, glycolysis and oxphos. And that's what I showed you previously. So in some ways, obviously it's got a few more bells and whistles than that, but it's really quite simple. It measures acid levels, it measures oxygen levels, and it can get you a picture of these complicated metabolic pathways. And here, as I showed you before, when you activate our NK cells, you ramp up metabolism. So you can see here, it's glycolysis. It's up into the energetic stage. You're using lots of oxygen as well for the citric manageable. So we asked, well, what happens if we add in a STREB inhibitor? There's lots of different STREB inhibitors we could use. We use 2 or 3 different STREB inhibitors. They all work in different ways. We also used a transgenic approach to knock out in transgenic mice genes that are required for activation of STREB within NK cells. So we really approached this from a pharmacological and a genetic approach. And no matter what we did, if we inhibited STREB, we completely blocked the ability of these cells to ramp up their metabolism. So no longer, we basically turn that switch off, and if you can't turn that switch off, and if you can't turn that switch on an NK cell, you stay down here with very relatively low levels of metabolism. And when that happens, their ability to kill tumor cells is severely impaired. They can't make cytokines, they can't make that cytotoxic poisons that they use to kill tumor cells. So this is very, very interesting. I'll show you some data with that in a mouse model of melanoma in a second. But this is, seldom is it as simple as that. It's never, we have one switch, to turn on the engine. Often there's multiple things. So certainly, STREB is really important for activating the configuration. So if you activate STREB, you should turn on the citric manageable that's required for elevated metabolism. But there's also other things. There's also the engine size. So for instance, another key switch that ramps up the engine size, so in terms of you make more mitochondria. So if you activate an NK cell, they almost double the amount of mitochondria they have. That's not controlled by STREB. They also ramp up all the machinery they require to metabolize glucose, and that's controlled by another transcription factor called c-Myc, which is less of a surprise because c-Myc is important in a lot of cells that are engaging in lots of growth. It's an important oncogene in a lot of tumours. So this is a much better known regulator of glucose metabolism. Well, so in the last 5 years, we've described these two things. We've used inhibitors of both of these. We've knocked both of these out, and using transgenic approaches. And if you inhibit either of those, you affect this metabolic increase, and you negatively impact their ability to find and kill tumours. So here's a bit of data to show that. So here's an approach we use to turn off MYC. So if you take away glutamine, glutamine is an amino acid, and MYC expression is really sensitive to glutamine deprivation. So here's what we call a Western blot. If you're not familiar with Western blots, the more of a black blob you see essentially is the more of that protein that's there. So here's our NK cells. This is done in duplicates, so there's two different samples done at the same time. So you can see a nice big black blob. I mean, there's lots of c-MYC there, so that switch is turned on. You see, and here you see when you take away glutamine for just a few minutes. This is 20 minutes. If you take away glutamine from the cell, they turn MYC off. MYC disappears. So the switch is turned off. And what happens if you do that? Well, here's your tumour cell killing. So cytotoxicity, this is where we've stuck our NK cells with a tumour cell, and we ask whether the NK cell can kill the tumour cell. But if you turn MYC off by taking the glutamine for half an hour, their ability to kill tumour cells is completely lost. Or just about not completely lost, but greatly diminished. Same for if you add an inhibitor of SHREB. So this is 25-hydroxycholesterol. It's basically a metabolite of cholesterol. It happens. It's found in a lot of situations in our body. It's an oxidized form of cholesterol, and it's a potent inhibitor of SHREB activation. So if you add 25-hydroxycholesterol, you turn this switch off. Here's showing that this is actually quantifying us turning that switch off. So SHREB is a transcription factor. This is looking at some of its target genes, and the expression is completely turned off. So it's a really potent inhibitor of SHREB activation. So here we are turning the SHREB switch off, and again we see a dramatic decrease in tumour killing ability. So when we put these NK cells with the tumour cells, they can no longer kill. So I think what we found is, in terms of going back to our car analogy, is that when an NK cell is really dependent on glucose. Because it does the citric molecule, glucose is pretty much its primary fuel source, and we found these two key switches, SHREB and MYC. And if you turn these off, they really struggle to kill tumour cells. So we wanted to test this in a real tumour model, so and in particular in an immunotherapy kind of model. So we decided to look at an NK cell-based therapy where we used a mouse model and we introduced NK cells as a way of therapy to see whether these switches are important. So this is how it works. You give the mouse a model of melanoma cells, and then you give them 3 therapeutic injections of expanded and activated NK cells, or else you give them just a mock injection. And you can see when they got NK cells where SHREB was turned on, you can see a very nice therapeutic effect. So this is just the size of the tumour growing over time, over 15 days. You can see here, when we don't give many NK cells, the tumour gets bigger and bigger and bigger, as you would expect. And when we give them therapeutic injections of NK cells, we can really have a real nice therapeutic effect, and this tumour stays much smaller. But if we give NK cells where we turn SHREB off, we completely lose a therapeutic effect. So this switch is really important for the ability of NK cells to provide a therapy against tumour cells in this case in a mouse. This is the size of the tumours at the end of the experiment after 15 days. Here's the mice that received no NK cells, or NK cell therapy with SHREB on, or NK cell therapy where we turn SHREB off. So it's very clear. It's important. Okay, so well, I think what we found out is that in terms of understanding how NK cells work, we know that NK cells have a slightly different and unusual metabolic engine, as it were, that has a citric molecule that's primarily fueled by glucose. We've got these 2 key switches called SHREB and c-MYC that have to be turned on to allow these cells to have potent killing ability or cytotoxic ability. So if we now know a lot more about it, but is this broken? So is this explaining why some of these NK cells in cancer patients aren't working well, or in other sort of patients that are more susceptible to cancer, like in obesity, for instance? So we actually looked at obesity because we know that obesity is now overtaken smoking as the largest preventable cause of cancer, and we, in collaboration with Dr. Lydia Lynch in Trinity, Bill Moshe, who's a clinician in St. Vincent's Hospital, he's an advisor to the Irish government on obesity, and Andy Hogan. We took cells from patients who were either obese or lean, and we did some metabolic analysis after we activate the cells. So again, this is what happens in normal. So taking NK cells from the blood of anybody here, we'd expect a nice metabolic response when we activate them with these cytokines. So here you can see overnight activation with cytokine, and normal NK cells ramp up their metabolism, become very energetic in their metabolism. But we took NK cells from people with obesity, and this you can barely see it. This is a change from here to here. They don't go around metabolically paralyzed. And patients alongside this data, we had a whole amount of functional data as well, which they did manually generated, showing that in these obese patients they have less NK cells. The NK cells don't make cytokines. NK cells fail to kill tumor cells. They've got really nice images of an NK cell beside a tumor cell, and they just can't even recognize them. They can't even polarize towards them, and are unable to kill them. And this is associated with a severe metabolic defect. So in obesity, it's very clear that the NK cell metabolism is severely impaired. We've also looked at collaboration with Claire Gardner, who's a colleague of mine in the School of Biochemistry and Immunology in Trinity, and her PhD students, Karen Slattery, at patients with breast cancer. And this is NK cells taken out of the blood of breast cancer. So we're not talking from within the tumour. This is peripheral NK cells. And again, we have evidence that here's from controls to healthy patients, nice metabolic response in response to cytokine. But these patients with metastatic breast cancer, we have severely impaired metabolism. And this is just one piece of data, but we now know that the mitochondria are looking very funny and dysfunctional. So there's lots of structural reasons why their metabolism is altered. So it's clear that in patients, the NK cell metabolism can be dysfunctional. It doesn't work as it should, and this is associated with a functional defect, with an impairment in their ability to make cytokines, an impairment in their ability to recognize and kill tumor cells. So it looks like the metabolic machinery of NK cells in obesity and in cancer is broken. There's other examples in the literature now in colorectal cancer as well, and liver cancer that confirms this. So, well, based on what we've learned about the metabolism of NK cells, can we think of ways to restore the metabolism, to fix the metabolism, fix the metabolic machinery as a way to restore their ability to find and kill tumor cells? So, in terms of that, we really need to know where and why. Where and why is it broken? This is a huge importance. So we can't fix things if we don't know why it's broken, and so this is something we're obviously working a lot to understand, and there's probably lots of reasons for it. So, for instance, we know that tumor cells use glucose as their main fuel as well. They love glucose. This is a PET scan. This is the way that they often diagnose tumors. Is they give you a radio-labeled fluorodeoxyglucose. It's basically a glucose tracer, and this goes to obviously goes to places the liver as well, but it lights up tumors. Here's some lung tumors. You can see a really bright signal. So that's because these tumors suck up the glucose because it's really important fuel. And because the tumors use glucose so much, and they take up so much glucose, often within a tumor there's very little glucose left over. And this could be one way that you get impaired NK cells within that tumor microenvironment, because there's no fuel left over for the NK cell. That's going to impair the whole metabolic machinery. So basically, you take the fuel away, the NK cells don't work. And we've done lots of experiments in vitro to show that. Decrease glucose concentration, or stop playing around with the fueling of the NK cell, and they can't kill. Well, what about these switches? So maybe the tumor is turning the switches off. So glutamine is also, because some tumors also use lots of glutamine as well, and they suck up glutamine. So could glutamine also be limiting within these tumor microenvironments? If there's no glutamine, as I showed you, in NK cells, MYC gets turned off within 20 minutes without glutamine. So maybe low levels of glutamine within these tumors is another way of turning the NK cell metabolic machinery off. In terms of turning off strength, well, oxysterols actually, it's become very clear that the tumor microenvironment is quite a high cholesterol environment. There's also evidence that there's lots of oxysterols. There's specific enzymes that make oxysterols. This is 25-hydroxycholesterol. We've also worked on 27-hydroxycholesterol, which is just as potent as an inhibitor of STRAT. A lot of the cells that are associated with the tumor microenvironment is is now becoming clear that they express the enzyme that make this. So tumours can make these oxysterol, 25-hydroxycholesterol, 27-hydroxycholesterol. We know that these turn off NK cells. If we put a cell that makes oxysterols and an NK cell in the same dish, the NK cell gets turned off. So we know that this is a way of communication between cells. So this could be another way that tumours are turning off the NK cell response. The tumour making these oxysterols to turn off this switch. And this is something we're actively working on and understand this based on a wide range of types of tumour cells, but certainly it's clear that this is a mechanism that can be used for certain tumours. Well, another thing that's often present at high levels within tumours, for various tumours, is anti-inflammatory cytokines. Cytokines like TGF-beta. This is so I've talked to you about some of the cytokines that NK cells make that try to ramp up the immune response. This is a cytokine that often dampens down the immune response, and we've shown that this cytokine, which is known to be quite high levels in cancer, can directly inactivate NK cell metabolism and turn off metabolism. So this is coming back to some of our CRS data again here. These are human NK cells, unactivated, activated, nice and energetic. If you activate them in the presence of TGF-beta, you can see you dramatically impair their metabolism. So some of these cytokines that are present within tumours are released by tumour cells and tumour-associated cells, can also act as a brake on the NK cell metabolic machinery. So if we start to understand how this works and the possible ways that they can be inactivated in the context of cancer, can we design ways to improve this, to make our NK cells bulletproof, as it were, to make them resistant to these conditions that can inactivate these switches? How can we improve our therapies? How can we improve our NK cell immunotherapies? So basically what we want to do is, one thing we might want to do is to make the NK cells within the cancer patient work better, or we may want to make sure that the healthy cells we put into the cancer patients don't start to lose their function once they're within the cancer patient. So basically we want to give them the NK cells a robust and irrepressible metabolism so that they can't be inactivated by the various conditions that are present within the cancer patient. So how might this be? Now this is obviously all very much ongoing. We have no clear answers yet. We have lots of ideas that we're chasing. So for instance, you may want to override this. So even in the presence of high levels of cholesterol, high levels of these 25-hydroxycholesterol, 27-hydroxycholesterol, that shred can't be turned off. Okay? And there are ways to do that. There's a version of shred that we can make that is as insensitive to oxysterols. It will always be turned on. Therefore, a tumour can't turn it off. Well, that would be useful, and we are generating those tools to test those at the moment. And we can try to think of strategies to increase the levels of gluten within the tumour microenvironment. And there's a few different ways you can think about doing that. Could we also try and increase the level of glucose fueling? And that might be something as simple as combining in an intelligent fashion the old-fashioned cancer therapies with the new immunotherapies. So for instance, how do we combine chemotherapy, which are basically just poisons that are designed to kill tumours quicker than they kill the patient? Can we incorporate traditional chemotherapy to try and shrink the tumour so that they're using less glucose, that there's more glucose available for when you come in with an immunotherapy, which is an NK cell immunotherapy, so that there's more glucose available when the NK cells arrive? Can we think about logically about how we can combine different therapies in this way? And of course, we can think about can we block TGF-beta? Can we use strategies to block the receptor, to block the cytokine? And would this be a way of maintaining NK cell activity? Certainly, if we take those NK cells out of breast cancer patients and activate them with cytokine in the presence of an antibody that blocks TGF-beta, we can restore some of their function, we can restore some of their metabolism. So certainly this does seem like quite an exciting pathway forward, blocking this cytokine that's able to turn off NK cell metabolism. So I think, coming back to this, I think these are all important. So in different patients, the reason why their immune systems might not be working well to eradicate cancer, and that's why they have cancer, because their immune system has failed. It can be different in different patients. We know from checkpoint inhibitors, in some patients, if you just take the brake off, that's sufficient to drive a really robust and strong anti-tumor immune response that can eradicate the tumor. In other patients, it doesn't work. So we've got to think about this whole picture. And we need to, in a patient basis, we need to understand why the immune response isn't working, and what do we need to tweak in order to drive a robust anti-tumour immune response. And that certainly involves understanding what are the key switches that are driving metabolism, and whether they are being activated in the patient, and can we engineer, so in those therapies I told you about where we're engineering cells in the lab, can we engineer them so that they have an irrepressible metabolism that can't be turned off? So I'd like to leave it at that. I'm happy to take any questions. Of course, I don't get in the lab anymore these days, but I have a great team of people who are allowed us to make all these exciting discoveries. I get to be that just curious person. I just think of experiments in the office. I run away to one of the guys and say, let's run this experiment and think back the results. So it's a great job also. And I've been really blessed. I've had some great people over the years who've gone on to do bigger and better things, and a current bunch of great people as well. Great collaborators as well that have allowed us to look at patients with cancer and understand why their immune systems are working, and also obesity, and of course some we couldn't do any of it without funding as well. So listen, I hope I pitched at the right level. I hope it wasn't too simple. I hope it wasn't too complicated, and I hope you've got something out of it. So my name is David Finlay, and thank you for listening.
I've got a question about the mouse model, SRABP.
Yes.
The mouse models. From what I can see from the slides, was it like a transgene kind of mouse model to turn on and off SRABP?
Turn on. So SRAB is a very complicated transcription factor.
Yeah, but as in, how did you generate the mouse model? Was it a transgene?
Yes, so basically, what we did is, so it's activated to a complex system where it needs to be translocated from the Golgi to the ER. It gets cut in two different places to generate the mature transcription factor, which then goes to the nucleus.
Okay.
Now there's a protein called SCAP,
Okay.
which basically chaperones it from the Golgi to the ER. Without SCAP, it can't go to the ER, it can't be activated.
Yeah.
So what we did, we deleted SCAP.
Okay,
So you delete SCAP, then SRAB is still there, but it just can't be activated. You can't generate the mature transcription factor.
that's a very drastic switch on and off of SRABP. But do you think that's actually achieved physiologically? Like, is it physiologically relevant to such a drastic on and off of the protein itself?
Yeah, so certainly, in terms of oxysterol, we can use levels of oxysterol that are similar to levels found in vivo. We can take, for instance, a macrophage, which is activated. They express an enzyme called cholesterol 25-hydroxylase that generates 25-hydroxycholesterol. If we stick an NK cell next to a macrophage, now we're not doing any transgenics, NK cell doesn't activate. And the shred targets are all down. So just having it in the same location of a cell that's making something that is targeting that shred does turn it off. So I do think it is happening in vivo. But yes, but transgenics allow us to do it in a cell-type specific way to tease it apart. Sometimes it is more, it does seem quite drastic. That's why it's always really important to combine it with different pharmacological methods. And then the whole picture starts to become clear.
And sorry, one last question.
Yes, of course.
Do you actually have any like transcriptomic evidence of these like NK cells in actual patient tumors or patient samples that show that this pathway is actually downregulated in terms of expression of RNA, for example?
So people haven't we haven't done that. No, we haven't. But we have got some functional data. If we take cells directly out of patients, we can see that they're, even before we do anything to them, their mitochondria are looking different, and their glucose utilization is different. So they are metabolically changed. We do know that things like so a molecule I didn't talk about today, which is linked to the shred, is mTOR. I don't know if you're sticking for it, but mTOR is upstream of shred. That's altered in these cells as well. So that we're accumulating evidence for that. But it would be nice to do it in more detail. But we're getting there. So we've done a lot of our mechanisms in mouse. I think we're now needing to move towards humans. To be honest, humans are great and all that, but I would prefer working. They don't want back. So we need to do that. We're looking for collaborations to do that in a bit more detail, which you're absolutely right.
Can the NK cells recover from the change in metabolism?
So that's a very good question. Certainly, when we take the NK cells out of breast cancer patients and we then give them cytokine in the presence of antibody that blocks TGF-beta, we can make them make more interferon gamma, so that we can start to recover some of that function. And those experiments are ongoing to see whether we can actually get them to kill better. So there's definitely a bit of reversibility of it. Absolutely, they're not completely exhausted, as it were. So that's one arm of it. Can we get them to metabolic recover so that they are better? But the other arm is then in all these therapies where people are, lots of different ways, people are trying to generate NK cells in the lab that you can put in. Can we make it so that they don't, once they get into that environment, obviously somehow detrimental to metabolism, can we make sure that they're bulletproof and they don't get turned off? But there's certainly some evidence that we can sort of rescue that altered metabolism from those patients, those cells coming out of the patients. But that's all.
Right, so you compared the NK cell loss of activity between breast cancer patients and obese patients. Do you think that it's the same mechanisms by which the NK cells activity is downregulated in obesity as it is in cancer, or what that was?
It's not the same, but it's probably similar. So we know that, for instance, this molecule called mTOR is down in both of them. In obese patients, SHREB activity is also impaired. In obese patients, probably a different trigger. So the NK cells in obese patients are really quite striking. They're packed full of lipids, so they basically take up loads of fatty acids and they've got all these lipid droplets within the cell. And that seems to be activating different receptors that are then turning off these signaling pathways. So it's all culminating in similar defects in the signaling. So SHREB's turned off and mTOR's turned off, but it's probably different triggers. We think in the breast cancer patients, it could be TGF-beta, which is driving the signaling pathways to turn off these switches, whereas in obesity it seems to be actually uptake of lipids is interfering with the signaling pathways and driving similar defects. So it's probably similar but not the same.
I have a question. So in the breast cancer patients, NK cells, did you mention that they are peripheral NK cells?
Yes.
Whereas a lot of the mechanisms that you described to like downregulate metabolism are like tumor microenvironment.
So even in the periphery, in people with breast cancer, they have elevated TGF-beta, and they also have elevated 27-hydroxycholesterol systemically, so even in the blood. But we also think that we're starting to get data that the NK cells have themselves undergone a process in the patients, and they are now starting to make TGF-beta themselves. So if you take the NK cells, purify them, so they're now away from all the conditions within the patient. You can still rescue some of their function by blocking TGF-beta. So the NK cells, for whatever conditions they be in the patients, whether it's traveling through the tumour or traveling close to the tumour, they've been altered so now that even the NK cells are making TGF-beta, which they can make and then turn off the metabolism themselves. So they've been altered by the environment. But there are multiple. That is the interesting point. So there are multiple mechanisms through which the tumours can affect NK cells at a distance. So at a distance, so even NK cells that are nowhere near the tumour can be affected through the production of these cytokines and these sterols, oxysterols that can act at a long distance in the periphery.
Any more questions? Yes, please.
In particular, reference to the natural killer cells research, but just generally, what is it that has prevented these breakthroughs to be made, say, 3-4 years ago? Are there any technical or theoretical advances that you think were key?
Well, I think that there's been massive breakthroughs in the last 10 years, but the checkpoint inhibitors are brilliant. So that kind of opened the door. This is the proof that people have been saying for a long time that you can get the immune system to do the work for you. You don't need to be using chemotherapeutics. They all obviously they have their place. But for a long time people were sneering, the immune system can't be used to treat cancer. But the checkpoint inhibitors prove that. And that's really opened the floodgates. Why aren't they? So they're in trials all over the place. So NK cells have been used for lymphoma. The CAR T-cell therapies have had really great success in treating specific types of cancer, in particular B-cell lymphoma. Great effects. It's not an all-in-one therapy. It's not an all-for-one therapy. So the problem is that every single cancer is different. So one therapy is not going to cure everyone. Same with the checkpoint inhibitors. 15% respond well. But why don't the other ones respond? Because they're different cancers. Even different patients with the same type of cancer, the tumor cells are different. So they don't all respond. That's why cancer isn't a disease. It's it's a combination of thousands of different diseases, but every single cancer patient comes with a different set of mutations, different set of reasons why that cancer formed, and that's why they don't all respond to the same therapy. But in terms of cellular therapy, it's very expensive, very expensive. So and some might say it's not really a workable solution for cancer treatment for all. Those costs are coming down a lot, and with all the advances that are coming online, and in particular with the development of off-the-shelf therapies where they can be frozen banks of these cells, I think it is going to become workable. So I think they're getting there, but it does take a while. You've got to get all the technical expertise to come online as well. So the manufacturing of these these are living drugs, which are a whole new set of sort of considerations as compared to sort of your classical drugs. So there's a lot of those sort of boundaries that need to be sort of managed as well to get those things where they get to. Yes.
When you put NK cells back in, do they proliferate, or are they in largely a post-mitotic state and they just go do their job and then have a half-life?
They can proliferate, yes. So there's people trying lots of different ways of doing this. So there's people who are using NK cells that come from peripheral from a fetal cord. They're using NK cells that are being differentiated from some inducible pluripotent stem cells. So there's loads of different trials going on, but certainly I think a key finding is that the longer you can get the NK cells to persist in the patient, the better the outcome on it. So when there is a longer persistence, it's definitely associated with some proliferation as well.
In the patient.
In the patient, yeah.
So, cancer cells, a lot of times they have altered metabolism themselves. They prefer glycolysis, they have less oxidative phosphorylation. So there are some complementary therapies, as they described. People would use ketogenic diet, for example, use less carbohydrates in their diet and things like this. What would happen with NK cells?
Yeah, so I think, in terms of glucose levels, our bodies are very good at maintaining glucose levels, no matter what you do. So even on so the liver will pump it out. So I don't think that's necessarily going to affect there's a danger with the ketogenic diet. So people are always saying this. I think most scientists would say it's a bad thing to be on cancer because one of the biggest dangers of somebody's in cancer is cachexia. So you don't want to be going on a diet that's going to be making you prone to wasting disease, essentially. So I think in terms of glucose, I think no matter what you do dietary-wise, your body will maintain glucose levels. So I don't think it's not the whole body glucose you need to worry about. It is that sort of directed microenvironment around the tumours where glucose can come in. And I think you're absolutely right. Glucose do a very distinct metabolism, and the school of thought is starting to move towards tumours evolve in our body through microevolution because they are evolving to basically escape our immune system. When we get cancer, that is a tumour cell that has gone through all these changes to basically discover through a trial and error process the ways that it can evade our immune system. And there's a school of thought now that the type of metabolism that the tumour cells adopt is also a way of evading our immune system because they decide to use glucose as a primary fuel and lots of it because that's the fuel that the anti-tumour immune cells require. Almost all the activated anti-tumour cells, cytotoxic T cells, Th1 T cells, NK cells, glucose is the key fuel. So if a tumour can deplete that, maybe that's another tumour evasion strategy.
We have time for one more question.
Yes. Can this knowledge of NK cell metabolism be applied in treatment for autoimmune diseases? And if not, I also don't know if NK cells have any.
So there isn't as much of a role for NK cells in the pathogenesis of autoimmune diseases. But can metabolism be harnessed for treatment of autoimmune diseases? Absolutely. So, for instance, there's a really nice study, again a mouse model, but there's real great proof of principle. So let's say for instance, in an MS, a mouse model of MS, it's called EAE. In MS, the problem is, multiple sclerosis, is you've got too many effector cells, you don't have any regulatory cells. So in T cells, in immune system, it's always important to have the effector cells and then the counterbalance, the regulation to make sure it doesn't get out of control. The regulatory T cells are out of control, And one of the key inflammatory cells that is overactivated in MS is Th17 cells. So T cells are making a cytokine called IL-17. And they look at the metabolism of these two cells and they notice a clear difference. And in a nutshell, regular T cells take fatty acids and they take it from the environment. They scavenge it. So they basically take whatever fatty acid they get from outside the cell and they use that. Whereas Th17 cells, they build their fatty acids. So they basically use glucose for building blocks and they build all the fatty acids that they need. So they thought, well, if I add an inhibitor of the enzymes that are involved in fatty acid synthesis, surely the regular T cells won't care because they scavenge it, they use it on the side. Whereas the Th17 cells, they will care. So they use an inhibitor of the first enzyme of fatty acid synthesis, and they got a completely skewing of the system. So they got loads of Tregs, almost no Th17s. They changed the balance, and in this mouse model of EE, they got a dramatically delayed onset of EE and a much less severe disease. So there's an example for us being able to see a key difference in the metabolic pathways used by a beneficial cell and a detrimental cell. They were able to switch the balance. So there's another paper on Tregs in tumours. It just came out there last week by a group in Switzerland, Humboldt Chi. They found that the Tregs, which are in the tumours, they use fatty acids as a fuel as well. Actually, they love fatty acids. And of course, in the tumour, you don't want any regular T cells because they're turning everything off. And they found that they are particularly require fatty acids as a fuel. And they targeted that, and they specifically targeted those Tregs within the tumours, and that had a nice therapeutic effect in those models. So I think, yes, absolutely, there's lots of companies springing up that are thinking that, yes, targeting metabolic pathways could be a new way to shift the balance of the immune system for cancer,